Hello students, I hope you all are fine and enjoying. This is Yogesh Kumar, postgraduate teacher biology from Jawahar Navodaya Vidyalaya, Shimla, Himachal Pradesh. Today we are going to learn a very interesting topic, polymerase chain reaction, but with the help of innovative tools. In my lesson, I am going to use first Microsoft PowerPoint presentation, second Lab Exchange, a free online platform for science education from Harvard University, third a PCR flash media player file which I have downloaded from Donnell DNA Learning Center, online mind mapping tools we are going to use, and last Wakelet for the curation of content. I hope you will enjoy the lesson. First, we are going to start with what is DNA. The full form of DNA is deoxyribonucleic acid. And where this DNA is present? DNA is present within the cell. Now we move to our lesson. What is PCR? PCR we are going to study polymerase chain reaction. This is the objective of my lesson, that is to provide the detailed information about the process of PCR. So what is the full form of PCR? Polymerase chain reaction. And it is a molecular biology technique which is used to make many copies of a DNA fragment. Now this polymerase chain reaction, it was originally developed in 1983 by the American biochemist Carey Mullis. Now, 5 core ingredients that are required to set up a PCR. First is the DNA template to be copied. Second, primers. Third is DNA nucleotide bases. Fourth is tag polymerase enzyme. And fifth is buffer to ensure the right conditions. PCR involves a process of heating and cooling that is called thermal cycling, and this thermal cycling is carried out by a machine. Now we are going to study the steps of PCR. First step is denaturing. When the double stranded template DNA is heated to separate it into 2 single strands, this step is called as denaturing. Second step is annealing. When the temperature is lowered to enable the DNA primers to attach to template DNA. This step is called as annealing. After that, third step is extending. When the temperature is raised and the new strand of DNA is made by the tag polymerase enzyme. This step is called as extending. Now, these 3 stages are repeated 20 to 40 times, doubling the number of DNA copies each time. Thank you. This is the Lab Exchange platform of Harvard University. I have designed a pathway in this platform about the PCR, and this pathway consists of 3 items, and straightway we can go to the pathway. Now the first step in this pathway, I have incorporated the written text material about the polymerase chain reaction. What is polymerase chain reaction? That is explained in detail how does PCR works, and what are the steps of PCR that I have included in the first step of the pathway. Now, after that, second step of this pathway is a video. Let's the video. After that, again, in the third step of this pathway, I have included a third video which is very clearly explaining the steps of. That video I have taken from Khan Academy. I hope you like the steps of this pathway, which I have designed on this Lab Exchange platform. Thank you. Now we are going to study polymerase chain reaction with the help of a flash media player. Now, amplification is given here. Now we enter for step-by-step study of polymerase chain reaction. Cycle 1, denature DNA. A PCR reaction starts with denaturing steps. Sample are heated to 94 to 96 degree for 1 to several minutes to denature the target DNA. We click here, denature target. Now, this is the denaturation step. After that, the temperature is lowered to 50 to 65 degrees Celsius for 1 to several minutes. This allows the left and right primers to anneal to their complementary sequences. The primers are designed to bracket the DNA region to be amplified. Now we click this tab, anneal primers. Now you can see downstream primer, and here you can see upstream primer. After that, the temperature is raised to 72 degrees Celsius for 1 to several minutes. This allows the tag polymerase to attach at each priming site and synthesize a new DNA strand. Now we click Extend Primers tab. Now you can see the tag polymerase enzyme, and that carried out the extension. Now we go to second cycle. Again, the denature DNA is the first step, anneal primers is the second step, and extend primers is the third step. So we can move to third cycle, denature DNA, anneal primers, extend primers. And we can go for the repetition of previous steps. Thank you. I hope you like the prime Now we are going to learn a very informative tool for making mind maps, because this is very beneficial tool for presentation of mind maps, presentation of content. I am using inforapid.org for making online mind maps. Now when I press this create a new user, a window like this it will appear. When I press continue, again continue, a window like this it will appear, and with the help of this tool we are going to make mind map of polymerase chain reaction. Now on the left hand side you can see very small windows. That edit diagram option is there, format diagram option is there, items in the category, memorize, notes, adjust view. Now first of all I am selecting the layout of my mind map, and I have selected the radial one which is appropriate for PCR. Now when I press this format window, you can see the color scheme and all the things, coloring patterns I can change. Now on pressing edit diagram, a new option appears. When I click this, then I write PCR. First I can write down the description of PCR here also, polymerase chain reaction, and I can also add the hyperlink. So many options are there. Now I will press OK. Now when I have pressed OK, again this diagram appears in the center of this platform. Again I can edit, I can format my diagram. Again I can add first step of polymerase chain reaction, that is denaturing. Again add the description, hyperlink of this. Now when I have pressed this is the second point that is attached with the PCR, and this shows the first step, denaturing. Again second step I can add. Second step is annealing. Okay. This is the annealing. Again, I can add the third step, that is extending. Now, this is the third step, extending. I can further proceed. I can edit. I can delete the item. Now we go for this 3D. Now I can rotate like this. PCR is the polymerase chain reaction, and these three steps are just rotating in a very beautiful manner. This is the beauty of this tool. Again, I can stop, just I can search each step on the web also. I hope you all liked this tool. We can download the image of this mind map in our PC also. And another point about this is memorization. We can memorize this. Now we are going to learn a very interesting tool, that is Wakelet, for the organization of content, for the curation of content which we have developed, the notes which we have developed, we can save on the cloud in the wakelet.com. Now I have prepared a wake polymerase chain reaction. When I click this wake polymerase chain reaction, now it will appear like this. We can change, we can edit the photograph, edit collection. We will go edit collection. We will change the image, edit the image. Now we can write down here about this wake polymerase chain reaction. After that, you can see these plus signs. Now, when you click this plus sign, there are so many options. You can paste URL here and you can save the URL. You can add the text also. You can add the YouTube links. You can add the tweets, bookmarks, images. You can upload the PDF. You can upload the links from Drive, from OneDrive, etc. Suppose I wish to upload the video from the Flipgrid, then I have to press this symbol. Now I am going to upload the PDF. I have clicked this upload PDF. Now this is the PCR, and when I open this link, this PDF will be uploaded. Here again, same plus symbol appears. When I click this and I wish to add something about the polymerase chain reaction, polymerase PCR, done. In this way, this tool provides the opportunity to curate the content, organize the content the way a learner wants. I hope you liked the tool Wakelet. Thank you.